Hello students, I'm your regular anchor for this course AMP 308 Agricultural Biochemistry. Today we'll continue from where we stopped. In our previous class, we talked about classification of carbohydrate metabolism, where we discussed the metabolic processes of glycolysis down to its end product, pyruvic acid. And we briefly talked about the end result of pyruvic acid or pyruvate, that in the absence of oxygen, pyruvic acid is converted to lactic acid. In the presence of yeast, pyruvic acid is converted to ethyl alcohol, while in the presence of oxygen, pyruvic acid precedes the TCA cycle, where it is acted upon by pyruvate dehydrogenase complex, which undergoes oxidative decarboxylation, converting pyruvic acid to acetyl-CoA. Today, we'll continue with our class and we'll look at the TCA cycle or Krebs cycle, the hexomonophosphate shunt or the PPP, pentose phosphate pathway. We'll also look at glycogenolysis, glycogenesis, and gluconeogenesis. I will wish that you will participate very well and be very, very active in this course, so that you play your own role as well. And I will expect us to have some minor exercises at the end of the course. Thank you as we move along with the class. Well, we are going to start now with our first topic, tricarboxylic acid cycle. Like I told you earlier on, I said in the presence of oxygen, pyruvic acid or pyruvate precedes the TCA cycle. Here we're going to look at the TCA cycle. The citric acid cycle, also known as TCA cycle, tricarboxylic acid cycle, or the Krebs cycle, is a series of chemical reactions used by all aerobic organisms to release stored energy through the oxidation of acetyl-CoA derived from carbohydrates, fats, and proteins. In addition, the cycle provides precursors of certain amino acids, as well as the reducing agents nicotinamide adenine dinucleotide hydrogenase (NADH), that are used in numerous other reactions. Even though it is branded as a cycle, it is not necessary for metabolite to follow only one specific route. At least 3 segments of the citric acid cycle have been recognized. The name of this metabolic pathway is derived from the citric acid, a tricarboxylic acid, often called citrate. The cycle consumes acetate in the form of acetyl-CoA and water, reduces NAD to NADH, releasing carbon dioxide. The NADH generated by the citric acid cycle is fed into the oxidative phosphorylation, that is the electron transport. The net result of these 2 closely linked pathways is the oxidation of nutrients to produce usable chemical energy in the form of ATP. In eukaryotic cells, the citric acid cycle occurs in the matrix of the mitochondrion. In prokaryotic cells, such as bacteria, which like mitochondria, the citric acid cycle reaction sequence is performed in the cytosol, with the proton gradient for ATP production being across the cell's surface, plasma membrane, rather than inner membrane of the mitochondrion. The overall yield of energy containing compounds from the TCA cycle is 3 NADH, 1 FADH2, and 1 GTP, that is nicotinamide dinucleotide, nicotinamide dinucleotide hydrogenase 3, 1 flavin adenine dinucleotide hydrogenase, and 1 guanine triphosphate. Here you see the cycle of the TCA, a complete cycle of the tricarboxylic acid cycle. This is another format of presenting the TCA for your own consumption, whichever comes to you for a better understanding, until its end product oxaloacetate plus acetyl-CoA plus water. It gives product of citrate plus CoH, and the enzyme catalyzing the reaction is citrate synthetase, and it undergoes aldol condensation. This is the same as 0, and this is the same as the step 0, and restart the cycle. The reaction is irreversible and extends to 4 carbon atom oxalocysteine. And with this, we now said the reaction continues in a cyclical form. That is why it is called tricarboxylic acid cycle. As long as the supply of oxygen continues, the cycle continues. Bioenergetics from glycolysis and TCA. The total ATPs that are produced in glycolysis is 8, the net ATP, and the various stages at which these ATPs are produced is shown in the table. And under the citric acid cycle, you have a total ATP of 30 plus the net ATP produced under glycolysis 8. So a total of 38 ATPs are produced. The total number of high energy phosphates compounds formed per molecule of glucose aerobically. Then total number of high energy phosphates compounds formed per molecule of glucose anaerobically is 2. Pathway, enzyme catalyzing the reaction, method of high energy phosphate production. You have glycolysis, glyceraldehyde 3-phosphate glycerate, 1 respiratory chain oxidation of 2 moles of NADH2. You have 2 times 3, you have 6. So the total net ATP produced from glycolysis down to TCA is 8. Compound of the respiratory chain electron transport system. ATP may be produced indirectly via the ETS. Most biological oxidations involve the removal of hydrogen ion from substrate, but the final combination with oxygen to form water occurs only at the end of series of reactions. A typical example is the removal of hydrogen coupled to nicotinamide adenine dinucleotide of the oxidation of isocitrate to alpha ketoglutarate. In this reaction, hydrogen removed from isocitrate is accepted by NAD and is then passed to flavin coenzyme. This donates 2 electrons to CoQ and 2 protons are formed. The electrons are then transferred via the sequential cytochromes to cytochrome A3, which is capable of transferring the electrons. The negatively charged oxygen finally unites with the 2 protons, yielding water. The reaction shown below. The ATPs are produced at the following sites between flavoprotein and CoQ, between cytochrome B and cytochrome C1, and also between cytochrome A and cytochrome 8, the pentose phosphate pathway or hexomonophosphate shunt. The pentose phosphate pathway is also called the phosphogluconate pathway, and the hexomonophosphate shunt is a metabolic pathway parallel to glycolysis. It generates NADPH and pentose 5-carbon sugars as well as ribose 5-phosphate, a precursor for the synthesis of nucleotide. Here you see the pathway of the PPP. This is another form of the pathway of the PPP. You see glucose being converted to glucose 6-phosphate under the catalysis of this enzyme hexokinase, and you see energy is consumed there because ATP is reduced to ADP. Glucose 6-phosphates generate 6-phosphogluconates, and the enzyme catalyzing the reaction is glucose 6-phosphate dehydrogenase. You see NADPH generating NADPH2. 6-phosphogluconates being converted to ribulose 5-phosphate. And ribulose 5-phosphates generates xylulose 5-phosphate, which is a 5-carbon atom, and ribose 5-phosphate, also 5-carbon atoms. And the ribose 5-phosphates is converted to pseudoheptulose 7-phosphate under the catalysis of this enzyme transcatolase. And the xylulose 5-phosphate precedes glyceraldehyde 3-phosphate. The xylulose 5-phosphates is converted to erythrose 4-phosphate, which is a 4-carbon atom. And this reaction is catalyzed by transcatolase. And the xylulose 5-phosphates that is converted to erythrose 4-phosphate, which is a 4-carbon atom, also leads the production of glyceraldehyde 3-phosphate and fructose 6-phosphate. The PPP continues. The oxidative reactions here, like we have discussed, is glucose 6-phosphate is converted to 6-phosphogluconate, and NAD is reduced to NAD pH and hydrogen ion. So at the end, the non-reactive stages of these reactions is ribulose 5-phosphate is isomerized to ribose 5-phosphate or epimerized to xylulose 5-phosphate. Ribose 5-phosphate and xylulose 5-phosphate undergo reactions catalyzed by transketolase and transaldolase that transfer carbon units, ultimately forming fructose 6-phosphate and glyceraldehyde 3-phosphate. Transketolase, which requires thiamine pyrophosphate, transfers 2 carbon units. Transaldolase transfers 3 carbon units. The entire set of reactions can be summarized as follows. You look at the table below. This is the summarized form of the reaction. Pentose phosphate pathway have some 3 major functions. The functions are shown here. The next is glycogenesis and glycogenolysis. Glycogenesis and glycogenolysis. Regulation of blood glucose is in close association with the biochemical regulation of glycogen pathways. Glucose 6-phosphates and fructose 6-phosphates, product of glycolysis, are linkers of glycolysis and glycogen pathways. Glycogenesis is the process of glycogen synthesis, while glycogenolysis is the breakdown of molecule of glycogen into glucose, a simple sugar that the body uses to produce energy. Here are the pathways of these two reactions, glycogenesis and glycogenolysis. Under glycogenesis, you can see the ATP, liver glucanase, muscle hexokinase, magnesium ion, glucose 6-phosphates being acted upon by phosphoglucomutase and converted to glucose 1-phosphate. The glucose 1-phosphate is acted upon by uridyl transferase, pyrophosphorylase. And in glycogenolysis, have a close look at it, you will see instead of glucose to glucose 6-phosphate, then to glucose 1-phosphate, here we have glycogen being converted to glucose 1-phosphate, then to glucose 6-phosphate, and two products are produced, the phosphatase and phosphohexoisomerase, leads to the production of glucose in the liver and kidney, and fructose 6-phosphate in the glycolysis, there in the skeletal muscle. Gluconeogenesis. This is gluconeogenesis is the generation of glucose from non-sugar carbon substrate like glycerol, lactate, pyruvate, and glucogenic amino acid. The vast majority of gluconeogenesis takes place in your liver and, to a smaller extent, in the cortex of the kidneys. Gluconeogenesis occurs during periods of fasting, starvation, or intense exercise, and is highly endogenic, energy intensive. Gluconeogenesis is often associated with ketosis, which is mostly observed in animals, and in most cases, pregnant animals. Some organs, such as brain, the eye, and the kidney, contain tissues that utilize glucose as their preferred or sole metabolic fuel source. During a prolonged fast or vigorous exercise, glycogen stores become depleted and glucose must be synthesized de novo in order to maintain blood glucose levels. Gluconeogenesis is essentially the reversal of glycolysis. However, to bypass the three highly exogenic and essentially irreversible steps of glycolysis, gluconeogenesis utilizes 4 unique enzymes. The enzymes unique to gluconeogenesis are pyruvate carboxylase, phosphoenol pyruvate, carboxykinase, fructose 1,6 biphosphatase, and glucose 6-phosphatase, because these enzymes are not present in all cell types. Gluconeogenesis can only occur in specific tissues. In humans, gluconeogenesis takes place primarily in the liver and, to a lesser extent, the renal cortex. Several tissues, including the brain, red blood cells, renal medulla, the lens and cornea of the eye, testes, skeletal muscles during exercise require continuous glucose supply. This is the gluconeogenesis pathway. Gluconeogenesis is the production of glucose from non-sugar precursors. Gluconeogenesis mainly occurs in the liver and involves the synthesis of glucose from compounds that are not carbohydrates. When a cell is growing on a hexose such as glucose and obtaining glucose for polysaccharides synthesis, there is no problem. But when the cell is growing on other carbon compounds, glucose must be synthesized. This process is called gluconeogenesis. Gluconeogenesis uses phosphoenol pyruvate, which is one of the intermediates of glycolysis, as starting material and travels backwards through the glycolytic pathway to form glucose. However, it involves several enzymatic steps that do not occur in glycolysis. Thus, glucose is not generated by a simple reversal of glycolysis alone. The major precursors of gluconeogenesis are lactate, amino acids, which form pyruvate or TCA cycle intermediates, and glycerol, which forms DHAP. The synthesis of 1 mole of glucose from 2 moles of lactate requires energy equivalent to about 6 moles of ATP. Pyruvate carboxylase converts pyruvate to oxaloacetate in the mitochondria. Oxaloacetate is converted to malate or aspartate, which travels to the cytosol and is reconverted to oxaloacetate. Phosphoenol pyruvate carboxykinase converts oxaloacetate to phosphoenol pyruvate. Phosphoenol pyruvate forms fructose 1,6-diphosphate by reversal of the steps of glycolysis. Fructose 1,6-diphosphate converts fructose 1,6-diphosphate to fructose 6-phosphate, which is converted to glucose 6-phosphate. Glucose 6-phosphatase converts glucose 6-phosphate to free glucose, which is released in the blood. Reactions involved in gluconeogenesis. 1. Conversion of pyruvate to phosphoenol pyruvate. In the liver, pyruvate is converted to phosphoenol pyruvate. Pyruvate produced from lactate, alanine, and other amino acids is first converted to oxaloacetate by pyruvate carboxylase, a mitochondrial enzyme that requires biotin and ATP. Oxaloacetate cannot directly cross the inner mitochondrial membrane. Therefore, it is converted to malate or to aspartate, which can cross the mitochondrial membrane and be reconverted to oxaloacetate. Oxaloacetate is decarboxylated by phosphoenol pyruvate carboxykinase to form phosphoenol pyruvate. This reaction requires guanine triphosphate, GTP. Phosphoenol pyruvate is converted to fructose 1,6-diphosphate by reversal of the glycolytic reactions. The second form of reaction is conversion of fructose 1,6-biphosphate to fructose 6-phosphate. Fructose 1,6-biphosphate is converted to fructose 6-phosphate in a reaction that requires inorganic phosphate and is catalyzed by fructose 1,6-phosphatase. Fructose 6-phosphate is converted to glucose 6-phosphate by the same isomerase used in glycolysis. Another form of reaction, which is the third, involves conversion of glucose 6-phosphate to glucose. Glucose 6-phosphate releases inorganic phosphate, which produces free glucose that enters the blood. The enzyme involved in glucose 6-phosphates. Thus, the net requirements to make one glucose molecule are 2 pyruvate, 4 ATP, and 2 GTP, 2 NADH, and 6 water. Thank you for listening.